moiety, which is soluble in organic solvent, or it is hydrophobic, moves into the organic phase. So here we are going to separate the drug of choice depending on its solubility. So the common solvents used for LLE extraction, we have ethyl acetate, trichloroethane, and rarely we are going to use ethanol or methanol. It depends upon the polarity of the compound. So suppose if the drug is moving into the aqueous phase, so we need to immediately separate the organic phase from the aqueous phase, and then we need to undergo centrifugation. Then the supernatant liquid can easily be injected into the HPL system if it is a aqueous soluble compound. Suppose if it is hydrophobic compound which has been easily moved into the organic phase. So organic solvents have a property of evaporation. So once they get evaporated, we can easily reconstitute the surface adhering drug molecule with the mobile phase, and then we easily inject into the HPLC. So this is regarding LLE method. So in LLE method, we are going to use invisible solvents, where we are going to extract the compound of interest, whether it is soluble in aqueous or organic phase. So this is the image of liquid extraction. And what are the ideal characteristics of this solvent employed in LLE is it should have low solubility with water, and should be highly volatile, and it should be compatible with HPLC detection, and the purity should be very high, and it should not cause any contamination to the sample. So these are the ideal characteristics of the solvent in LLE. Next extraction process is SPE, that is solid phase extraction. So here the principle of separation is absorption. So it is very much similar to your chromatographic technique. Here we are employing a sorbent, and sorbent we are going to allow the sample, this plasma sample, over this cartridge. Cartridge is filled with sorbent, and due to the affinity towards this sorbent, the separation takes place. So this separation mainly has 4 steps, that is conditioning step, loading step, washing, and elution step. So conditioning is, as I said, it is a cartridge, and inside the cartridge we have a sorbent layer, which generally made up of its silica gel. It's very much identical to the column of HPLC, and which is non-polar in nature. And we are going to condition this sorbent layer with the methanol first to remove any adhering impurities. So this process is called conditioning, and we are going to treat it mobile phase as well to adopt the cartridge to your sample. Then we are going to add sample, and you can see the different colors of the sample, which includes analyte plus interference. So sample is loaded by into the cartridge, and due to affinity towards its surface or towards the sorbent, the separation takes place. And finally, when the interference are being eluted out, we need to wash, which creates a positive pressure to elute the desired analyte with the help of mobile phase. So this is the 4-step process of the SP. And comparatively LLE method, this SP method is more reliable, but the cartridges are expensive. And the last method is protein precipitation. Here we are going to use protein precipitants like acetonitrile to the plasma. So addition of this acetonitrile to the plasma, the proteins get precipitated. Immediately we need to centrifuge it. Generally we are going to use cooling centrifuge at -4 degrees centigrade at 2000 to 5000 RPM range for 20 minutes. And we are going to collect the supernatant liquid, and this liquid is being filtered to avoid any particulate matter, and then it is being injected into the HPLC system. So these are the 3 methods of sample preparation: LLE method, SPE method, and protein precipitation. Once the sample is being free from interference, now it is ready for your method development. So for your convenience, today I am going to explain the bioanalytical method with my work, that is bioanalytical method and development and validation of eprosartan. So this drug has used, and one important thing is that whenever you are performing bioanalytical methods, we need to use internal standard. Unlike other analytical methods, regular analytical methods, where there is no requirement of internal standard, but in bioanalytical methods, we are going to use internal standard. So internal standard concentration should be maintained constant with increased concentrations of your sample. So in bioanalytical methods, we need to prepare separately calibration standards as well as quality control standards. So quality control standards should be prepared in the range of the calibration standards. So we need to prepare LQC, QC, and HQC. LQC, lower quality control standard, medium or middle quality control standard, and high quality control standard. So these are the three quality control standards should be prepared within the calibration range. So we have minimum number of calibration standards are 5. So we need to prepare more than 5 according to US FDA guideline. And parallelly, we need to prepare quality control standards, and internal standards should be added. So if you see the chromatograms, the drug is eprosartan, and internal standard we have maintained same concentration to all the calibration standards. So 0.3 microgram per ml, 0.6 microgram per ml, 2 microgram per ml, 3 microgram per ml, 5, 10, 15, and 20. So the purpose of internal standard is to compensate with the method. So this gives an information that the method is running well. So how to select the internal standard? Internal standard should be selected based on physicochemical properties and pharmacological properties similar to the drug, and there should be a minimum of 2 to 3 minutes distance of RT between the drug as well as the internal standard. So these are the ideal things to be noted down when we are going to select internal standard. Structurally similar, that is physicochemical properties, can also be taken into consideration. So calibration, once calibration standards have been injected into HPLC system, so it gives peak area, and the peak area ratio is being taken on y-axis, and on x-axis we are going to take concentration, and we need to construct a calibration curve and take it that calibration parameters like y coordinate, x coordinate, and y-intercept should be, and R-square value should be mentioned in the graph. So this is the method how we are going to prepare concentrations starting from calibration standard 1 to calibration 8. So we have prepared 0.3, 0.6, 2, 3, 5, 10, 15, 20 microgram per ml of the eprosartan drug, and we are going to add drug stock solution to the sample, and together we are finally make up to 1000. So 98 microliters, 976 microliters, 920, 880, 800, 600. So together it should be 1 ml, that is equal to 1000 microliters. So to prepare 0.3 micron per ml, we need to take stock solution, that is eprosartan aqueous 12 microliters, and we need to spike with 988 microliters of plasma. Here, aqueous phase indicates that blank plasma. So if you see the total concentration of individual, it reaches to 1 ml, that is 1000 microliters. So after seeing this table, you can understand that the peak area of internal standard is constant because we have injected same concentration of losartan to eprosartan drug, same concentration in all calibration standards. So there is increase in peak area of eprosartan as concentration is increasing. So whenever we are going to divide eprosartan with internal standard, we are going to get a ratio of peak area, which we are going to use this peak area ratio for constructing calibration curve by taking y-axis peak area ratio and x-axis concentration. So I think this method is clear for you, because the next step we are going to go for the validation part. So if you have any doubts in this part, you can Saravanan sir.
Sir, actually, to avoid the disturbances, we muted all participants, sir.
Okay, so finally at the end of the session we can.
Disturbances will come.
Okay, at the end of the
finally
session,
we can have an interaction session.
Yes. Because I will answer the final how to prepare this calibration standards. So coming to method development part is over. So we are going to deal with this validation. So validation, as I said previously, we are going to adhere to US FDA guideline, and I have recently gone through this new adaptation of ICH guideline M10. Final draft has been released on 24th May 2022, and it is going to upload to the ICH website from 21st January 2020. So it is very much similar to a regular US FDA guideline validation parameters. So it includes selectivity, specificity, accuracy, precision, recovery, matrix effect, calibration curve, carryover, stability of unlight in spiking samples, limit of detection, and limit of quantification. So selectivity. So selectivity, it is the ability of the method to differentiate and quantify the drug in the presence of sample. So here we are going to inject blank samples of plasma, which is procured from 6 sources, and we are going to inject into HPLC, and we are going to see that there won't be any there won't be any possibility of detection of this interference by the HPLC system. So for this, what we are going to do is, initially the drug is being injected, and we are noting down the retention time, and again we are going to inject blank samples, and we are going to see whether the RT is being affected by this blank sample. So this is selective. It is ability of the method to differentiate and quantify the drug in presence of sample matrix. So we are going to do with the 6 sources, 6 different sources of plasma. Second parameter is specificity. So specificity is the ability of the bioanalytical method to detect and differentiate the analyte from other substances, which may be structurally similar to the analyte, metabolites, isomers, impurities, degradation products formed during sample preparation. And we should, after finding this, that is after injecting the sample, the calibration standards, the responses detected and attributed to interfering components should not be more than 20% of the analyte response at LLOQ, and not more than 5% of internal standard response in LLOQ sample. So LLOQ is nothing but the lowest concentration calibration standard. So LLOQ is the lowest limit of quantification of the sample. So if it is detected, it should not exceed 20%. And the internal standard which is being added to the calibration standard 1 should not be more than 5%. So this is the criteria for passing this specificity. Accuracy, it describes the closeness of average test values, the true value of the analyte. So how close the value to the test we are going to find in this accuracy parameter. So accuracy should be measured using minimum of 5 determinations per concentration. As we have concentrations, 8 concentrations, we need to choose any one concentration and we need to inject it for 5 times. So the average value should be within 15% RSD and for all the concentrations, and except for LOQ, it should be not more than 20% RSD. RSD is relative standard deviation. So being a bioanalytical method, the RSD is being lax up to 20%. If it is a regular analytical method, it is less than 2%. So the deviation of the average from true value helps on the estimation of the accuracy. So generally, these accuracy studies are done in a single day with a number of times in a day. Now, precision, it is the closeness of individual drug responses when the method is applied repeatedly to multiple aliquots of single uniform volume of biological matrix. So this precision should be determined using a minimum of 5 determinations per concentration. So precision should be determined using minimum of 5 determinations per concentration. So we need to inject the selected concentrations for 5 times. So the precision at each concentration level should be not more than 15% of CV or RSD, except for LLQ, that is 20%. And this precision is further divided or classified into intra-batch precision or repeatability. So this intra-batch precision is done in a single day with a single person. So this measures precision during a single interpretation, single run, single person, and single day. So it should adhere to the same criteria, that is 15% of CV for LOQ for remaining concentrations, and for LOQ not more than 20%. And between run is nothing but the number of days. The number of days will be 2 to 3 days. And here interbatch precision or repeatability. Here we are going to change the analyst. We are going to use the equipment which is identical to the previously done HPLC or UV. Identical specification should be there, and laboratory conditions should be similar, and reagents, but they are not identical. So this is the types of precision we are going to carry out, and the criteria should follow. As I said, LLOQ should be not more than 20%, and other concentration ranges should be within 15% of RSD. Now recovery. Recovery is the detector response obtained from the biological matrix, which is compared to the true concentration of the food stamp. So recovery is the extraction efficiency of the determination within the limits of variability. So for this recovery studies, we are going to use only the quality control standards, that is LQC, HQC, and NQC. So here we are going to demonstrate the recovery studies by using these 3 determinations with unextracted standards, which represents 100% recovery. So here unextracted means drug in aqueous, and which are compared with the quality control standards. So these recovery studies are further subdivided into freeze and thaw and benchtop stability studies. So it is a function of storage condition, the chemistry of the drug, the matrix, and the container. So how any alterations are being found upon storage of this sample in a refrigerator or deep freezers. The stability studies should evaluate the sustainability of drug sample collection and handling after long term, that is in frozen condition, and short term, that is on benchtop. So we are going to do minimum of 3 freeze-thaw cycles. That is, we are going to withdraw the frozen samples and keep on the benchtop and leave it for some time to attain the room temperature. Again, we are going to get back into the frozen condition. So we are going to repeat the cycle for 3 times, and then we are going to analyze using HPLC or LCMS whether there will be any change in the matrix condition which is affecting the sample. Now, next is matrix effect. So the matrix effect is defined as the alteration of sunlight response due to interfering and often unidentified components in the sample matrix. So this matrix effect should be evaluated by analyzing at least 3 replicates of LQC and HQC. So each prepared using matrix for at least 6 different sources. For each individual matrix source evaluated, the accuracy should be within 15% for HQC, and for LQC it should be less than 20%. So this is the matrix effect. Limit of detection. So limit of detection is the lowest concentration of sunlight that can be detected but not quantified under experimental conditions. So this limit of detection can be easily related with the accuracy studies. So accuracy studies, I said, minimum for performing analysis require five concentrations, and which have been prepared in triplicates, and we are going to divide the day into 3, that is morning, afternoon, and evening, and we are going to average the morning 3 triplicates data, afternoon 3 triplicates data, and evening 3 triplicates data, and we are going to construct the calibration curve by taking average of the 3. So when you are constructing calibration curve, you'll get the calibration parameters like y coordinate, y intercept you will get, and you have slope values, and we are going to have concentration on x-axis and peak areas on y-axis. So to calculate this LOD, we are using a formula 3.3 into standard deviation of y-intercept divided by average of slope. So the standard deviation we are going to take as numerator standard deviation value of y-intercept and average of slope. And when you are doing this calculation, whatever the answer you are getting should be less than the concentration of the LLOQ, that is lower limit of quantification. The units of this LOD are microgram per ml or nanogram per ml. Whatever the units you are using for preparing calibration standards should be used for this LOD. So the SD is nothing but standard deviation of response, that is y-intercept, and this is the slope of calibration, average of slope of calibration. So this LOD can also be adopted for precision also, where we are going to do the studies for 3 consecutive days. And as I said, we are going to get 3 calibration curves per single day. So for 3 days, we are going to get 9 calibration curves. And from this 9 calibration curves, we need to calculate standard deviation for the intercept, which are going to take a numerator, and 9 slope values, which we are going to calculate as mean, which we are going to take as denominator. And finally, the value should be less than the first concentration, the calibration curve. LLOQ, limit of quantification, it is defined as concentration at and above which the analyte can be quantified. So, which is where the sample, where the instrument can be detects the sample concentration and gets immediately quantifies it. So this is the lowest level of detection, and followed by quantification by the instrument. So it can be calculated by using the formula 10 into standard deviation of y-intercept divided by average of slope. So same thing we are going to use as numerator here, the average of 9 y-intercept values and 9 slope values average, and the value what you are getting of LOQ should be less than LLOQ in the calibration or calibration standards. Next validation parameter is carryover. So carryover is an alteration of measured concentration due to residual analyte from a preceding sample that remains in the analytical instrument. So carryover should be assessed and minimized during method development. During validation, carryover should be assessed by analyzing blank samples after the calibration standard at the upper limit of quantification. So this method is very, very simple. Here what you're going to do is, whenever you have finished the calibration standards, immediately we need to inject a blank sample to prevent any carryover standard which has been in the instrument. So that may be interfering with your peak area. So to prevent that, we need to inject a blank sample followed by, and whatever suppose the blank sample has given any response, that should be not greater than 20% of LLOQ and not greater than 5% of the internal standard in the LLOQ. Now we are going to deal with robustness. This method is carried out to check the ability of the method to remain unaffected with small changes in the method parameters, which include temperature, that is column temperature, injection volume, that is normal injection HPLC will be from 10 microliters to 25 microliters. Any alteration, any increase or decrease in injection volume, and solvent system, that is mobile phase composition, any alteration in mobile phase composition, alteration in buffer concentration, any alteration in the pH, the addition of acid or base to the mobile phase. So that may have any effect on the response of the instrument towards the method. And method validation ruggedness. Ruggedness is not a part of ICH, but it is being adopted by US FDA, and it is defined as the degree of reproducibility of results of same sample under a variety of conditions, which include different laboratories, different analysts, and different environmental and operational conditions. So which is very much similar to precision repeatability, where we have seen that the same method being transferred to another laboratory, where the method is being adopted by different analysts on different instrument and different laboratory conditions. So there should not be any change in the method. Sir, Arun, sir.
Sir, tell me, sir.
Sir, it's we have concluded the last part.
Okay, sir. Just a minute. So if you have any queries, you can raise your hand. We will unmute you. You can directly interact with the speakers. Sir, thank you, sir. Sir, please unmute that Ramya. Yes, Ramya, you can ask your questions.
Yes, sir. Can you hear me, sir?
Yes, ma'am.
Yeah, thank you, sir. Thank you for unmuting me. My question is to sir. Very good morning, sir. Sir, while doing this bioanalytical method, how to fix this LLOQ and ULOQ with respect to Cmax of the drug?
Yes, it's a good question. The main purpose of bioanalytical method is to, your question is how you are going to fix the Cmax or how to prepare quality control standards?
Quality control standards with respect to the Cmax of the drugs, sir.
So
what we are studying.
So bioanalytical method is being adopted in two forms. One is for NDA and ANDA processing. So
Okay,
suppose
sir.
if you are going for ANDA, abbreviated new drug application, that is we are going to test only the bioequivalence study. So you already know the known drug and the Cmax. So once you know the Cmax, it's easy to prepare the calibration standards followed by quality control standards. For example, if you want to, suppose you have 8 calibration standards prepared, C1 to C8. If you want to prepare LQC, that is lower quality control standard, it should be 2 times to the first concentration. Is it clear? C1,
2 times of
C1, that is first calibration standard.
Okay.
Generally, we'll term it as LLOQ, lowest
Yes, sir.
limit of quantitation.
Yes, sir.
So 2 times. Suppose if you are preparing for MQC, MQC should be 60% concentration of the C1. 60% of concentration of C1, and HQC should be 90% concentration of C1.
Okay, sir.
So if you calculate, we can easily, and the simple method is regularly dividing first two concentrations by 2, then in that manner also you can prepare quality control
Yes,
standards.
sir. Actually, even I thought of asking you how to fix this quality control standards. Is there any method like first, if for example C1 is 1 microgram per ml, what would be the C2, C3, C4 like that on to
So
till C8
yes. The method what I said just now, 2 times of the C1. That will
of
be
each concentration 2 times?
2 times. Suppose C concentration has response on the example. You multiply it, double concentration will be LQC.
Okay.
60% of it will be MQC and 90% of it will be HQC.
Okay, sir. Thank you. And one more question is regarding this plasma is to drug is to internal standard ratio, sir. Is there any thumb rule that we should follow that plasma should be taken of this microliters with compared to drug and internal standard?
Yes, okay. If you go back to my slide, there is a chart. I have shown you a table. So I have taken, for example, I have made maximum amount of blank plasma. You can see the last table in
Yes, sir.
microliters, 988
988.
microliters.
Yes,
So
sir.
my aim is to have 1 ml. That is
On the
1000
whole, 1
microliters.
ml.
1 ml. So that is a, as you said, it's a thumb rule where we can
Okay.
fix 1 ml. So as 1 ml is very low concentration. So in
Okay.
this we are having more amount of aqueous part. That is blank plasma is much more. And you
Yes.
see only 12 microliters is our drug.
Yes, sir.
So we are increasing only drugs concentration and we are slowly increasing the blank plasma where aqueous phase is more.
Okay.
So this is, you can fix 1 ml as maximum for blank plasma, including
Oh,
spiking with the drug.
okay. What about the internal standard, sir, here?
Internal standard, I have used it 50 microliters.
Constant.
Constant. That is maximum you should not go beyond 50. So you can start from
Okay.
2 microliters to 50 microliters.
Okay,
If
sir.
it's any concentration.
Okay, and this is regarding if we are taking more like simultaneous estimation of 3 or 4 drugs, then how to take the drug stock solution for plasma? If I'm keeping plasma as 988 itself, the other 12 microliters should be divided into 3 if I'm analyzing 3 analytes means, isn't it, sir?
Yes, because you need to adjust the drug stock solution within that range only. You should not disturb the blank plasma.
Okay, sir. Okay, fine. Thank you so much, sir. It's very informative and it is very helpful. Thank you, sir.
Welcome.
Thank you, madam. Thank you for your queries, and thank you, sir, for answering that particular queries.
Thank you, sir.
Any other participants? If you have any queries, you can raise your hands. We will unmute you, and you can directly interact with the speakers. Or otherwise, you can place your questions in the chat box. We will ask. And Mani sir, in the meantime, there is a questions from that YouTube. What is that industrial opportunities in that bioanalytical techniques they are asking?
So you want to know if you have grip on these bioanalytical methods, what jobs you are going to get.
Yes.
So the basic thing is that you should be very much perfect in validation parameters. And just now the madam has discussed regarding the preparation of samples as well as how to construct calibration curve, how to dilute the sample or how to spike with the drug with the plasma. So these techniques, these are benchtop techniques which are very much essential for getting industrial job where we have wide scope in that. And to my knowledge, the new ICH M10 guideline will give and will solve many questions because lots of confusion was there, which method should be followed. And many of them, they are following USFDA guidelines so far till today. So this M10 guideline will give much information regarding methods in future.
Thank you, sir. And another question is from YouTube. Why sodium chloride is added before centrifugations?
Yes, very good question. Sodium chloride or NaOH, sometimes sodium hydroxide is also added. So we have we know that gets binded to the proteins. So that protein should relieve or withdraw the drug from its surface. So here the sodium chloride enhances it breaks the bond between the protein and the drug and makes it move towards the aqueous phase. that is your mobile phase. So that's a reason we are going to add sodium chloride in before centrifugation process. You know that we are going in LLD method. If you take invisible solvent, you are going to add. Then we are going to add a little bit of, as you said, NaCl is added and undergoing centrifugation. So this sodium chloride enhances the withdrawal or breaking the bond between protein and the drug. So where we can enrich the mobile phase with more amount of available drug.
Thank you, sir. And any other peoples, if you have any queries, you can raise your hands. We will unmute you. And the another question, sir. So, what precautions we should take while doing this bioanalytical method developments?
Precautions in what sense you're asking? In
So
instrumental part or or preparation part?
Both the cases, sir. So compared to
So,
that normal method development and bioanalytical method development.
Because this analytical method will be shifting to instrument. Suppose you are going for HPLC. HPLC, we are able to only quantify. We are only able to isolate the drug, and we are going to calculate according to the peak area. But suppose if it is a new drug application, here HPLC cannot give that much information, right? Because we need to go for structured elucidation. So if it is hyphenated with LCMS or tandem with any MS mass spectrometer, so it enhances wider information of the drug. And so people who are being interested in knowing about more, they should know how to handle the LCMS also. So for better knowing the possibility of this method in the future.
Thank you, sir. And one of the students asked, sir, whether there is any specific institute or training center is there for getting an what is that practice in that bioanalytical method development?
Yes, we have lots of opportunities in terms of training. If you see that DST has given strategic scheme, which has been recently, it is in ICT Mumbai. They have conducted this workshop for 15 days. So they are helping the students in gaining the knowledge and handling of sophisticated instruments like GCMS and LCMS. And it is till the date is on. People who are interested, they can they can apply, because from 25th to 30th, they are 25th to first week of September, they have extended the date. So DST is regularly conducting more workshops. So regarding the handling of these instruments. So, even CDRI is also giving training in NCL Pune, even ISCD Hyderabad, they are also involved in training with the sophisticated instruments, mainly dealing with the bioanalytical methods.
Thank you, sir. And even if you have any further queries, you can send your queries to our email ID, and we will forward to that speakers, and we will get back you to that answers. And Dr. Mani, sir, thank you for wonderful presentations on bioanalytical method development and validations. Particularly that your presentation was aimed with that latest version, which was updated on May 2022, which will be very helpful for the young research people, those who are pursuing their master degree and PhD in their analysis. For them, definitely it will be an eye-opening sessions, and they will get a huge knowledge from your presentations. So once again, I'm from the.
My suggestion to the young researchers is that try to do more pharmacokinetic part. Regular analytical methods you are doing, they're very good. That's enough. Regularly you are using aqueous and organic solvent mobile fluids you are doing, and regular analysis you are doing, simultaneous estimations. My advice is to try to go a little bit ahead in performing kinetic part. BAB studies, then we can have more scope in learning these validation parameters, especially my advice is to download this M10 guideline. All of you, all the participants, try to. It's freely available in the net. So MT guideline ICH, if you type in the Google bar, you will get this document. So the document is very, very useful for you to implement your further research. As Saravanan sir said that it will be useful for their research in PhD level or in farming.
Thank you, sir. Sir, can you stop your screen sharing, sir? Manish, sir.
Sir.
So now I'm requesting our principal, Dr. V. Alagarsamy, sir, to conclude the sessions and to felicitate our speaker.
Thank you, Dr. Manish, for your wonderful lecture. I hope all the audience have interacted very well and they have been clarified many of their doubts, and you have also answered in a very nice manner. And it's fortunate to have a wonderful lecture, and hope, because this is one of the field where there are much more opportunities students can explore, and we can get much employment also, those who are interested in the IM-Pharm analytical chemistry. And really it is nice. We have explained very well about the bioanalytical method development and the validation, and hope all the audience must have benefited very much with your inspiring speech. And with this certificate and the token of appreciation for your wonderful speech and your valuable time, kindly accept this certificate of appreciation and also the virtual memento for your wonderful speech and the valuable time you have spared with us. Kindly accept this, sir, and thanks a lot for accepting our invitation and doing this wonderful webinar. Thank you very much.
Sir, I want to add a small thing, sir.
Yes.
Sir, I have referred your book, sir.
Yes,
sir. So, which I am that practical inorganic chemistry. It is very much useful because I am referring it from past 10 years.
Okay, sir. Thank you very much, sir.
It is very useful to students because most of the experiments are designed very well. So I thought of meeting you many times, but I never found the opportunity. So
Okay,
I thought of thanking you because many of our college students and faculty are using your books.
Okay. That was the first book I have authored in 2009.
I have seen it has been much the books have been much sold also, because I have seen in many institutions. Whenever I visited the library, I have seen the book.
Okay,
the limited part has been very much effectively covered.
Yes, sir. All like whatever we are using in the regular class, we have prepared in the form of books and we have submitted, sir. So that's all. So hope it will be useful even our the next books like medicinal chemistry, organic chemistry, recently biochemistry, and the computer aided drug design also we have done. Biochemistry I have authored with Dr. Saravanan.
Sir. Congratulations and wonderful, sir.
Yes, thank you.
Because I have seen in the Flipkart and Amazon, your books have been available.
Okay, sir. Thank you for your compliments, sir. Thank you, Unandal. So I hope all the audience must have enjoyed the presence today's webinar. So with these few words, I thank Unandal for joining this webinar. Hope we'll have better webinars like this in the coming days also. Kindly join in the future webinar also. And also I wish to put a note on this gathering. We are organizing a PowerPoint presentation competition as we are celebrating the 75th independence year in this during this year. So we will also select plan to develop we plan to compete among the pharmacists and organizing a PowerPoint presentation competition, and it is organized for the throughout the country. So anywhere from Kashmir to Kanyakumari can participate and win. And our aim is to celebrate the 75th year of Independence Day as well as the 75th year celebration of the pharma industry development in this country. So let us all join for this competition and come together for the celebration of the 75th year celebration Independence India as well as the pharma developments. So thank you, Unandal, for joining this webinar. So I think if no other words, so Dr. Saravanan can sign off.
Thank you, sir. Thank you, sir.
Thank you. Thank you. So Dr. Jayaprakash,
So all participants are asked to fill your feedback forms. So you will receive your certificate within 3 days to your registered email ID. Thank you, one and all.
Dr. Jayaprakash, you can conclude.
Yes, sir. Thanks. Thank you, sir. Thank you very much, everyone, for participating and their cooperation to complete this webinar without any struggles. Thank you very much. Thank you, everyone.
Thank you, sir. You can sign up.